and welcome to our Week 2 lab video. This week, we will be reviewing some basic biochemistry as well as some cell structure. This video covers Part 1, Lab 2 Basic Biochemistry. This week's labs do require you to have your lab kit, so if you have not yet received your kit, please contact me immediately. After reviewing the pages 37 through 42 in your lab manual, your first experiment, we will use pipe cleaners and beads to model protein structure. Answer the questions as you go along, and if you have a smartphone or camera phone, take a picture of your progress to help you remember. For experiment 2, a full list of materials is located on page 46 in your lab manual. This is our first experiment that will require reagents. I will briefly review what these reagents and materials are. First, you will need graduated cylinders, 100 milliliter and 10 milliliter size. You will also need your test tubes in the rack. You will need two 250 milliliter capacity beakers. You will need 5 transfer pipettes. You will need to locate your reagents box and find Biuret reagent, 1% glucose solution, as well as your unknown solution. Experiment 2 will look at using the Biuret reagent to test for the presence of proteins. Biuret reagent is normally blue, and when it comes in contact with proteins, it becomes a purple color. Your task in this experiment will be to determine if an unknown solution contains proteins or not. Record your results on page 48 and answer the questions as you go. Experiment 3 will look at what effect changing the temperature has on the ability of an enzyme to do its job. Remember from your reading that enzymes are proteins and are definitely affected by the temperatures in which they are present. A full list of materials is listed on page 51 in your lab manual. Here I'm showing you what the 3% hydrogen peroxide bottle looks like. To review, the reaction that we are going to be studying is this one, where you have hydrogen peroxide gets turned into water and oxygen gas by the presence of the enzyme catalase, represented here by the purple. In our experiment, we will be using yeast as a living organism as a source for our enzyme catalase. And what we will be measuring is the oxygen gas production. To do that, you will use your balloons and put them on the ends of your test tubes. The circumference of the balloon that you measure with the string will measure essentially the oxygen gas produced by that reaction. Remember, the larger the circumference, the more gas that is present in the balloon. Record your results on page 54 of your lab manual and answer the questions as you go along. Continue on to Part 2 to review the Lab 3 Cellular Anatomy and Physiology.